Hello, class. My name is William Grant Hale, Jr. This discussion today is for the different isoforms of ADALase and their different expression patterns. This is in part for the fulfillment of the Biochemistry 530 class from Grand Canyon University. First on the introduction, this is what I'm going to be talking about today. I'm going to talk about the ADALase. Will be described as what it is, its history, specificity, composition, and molecular characteristics. And the three types of ADALases will be covered: Aldo A, Aldo B, Aldo C. And also we'll be talking about the ADALase gene expression as far as dealing with an example, the African clawed frog, the Xenopus laevis will be covered. So actually, what is ADALase? ADALase is present in all animal and plant tissues and in most microorganisms. Class 1 ADALase found in the animal and higher plant tissue is characterized by the lack of requirement for a bivalent metal cofactor. In Class 2 ADALase, found only in prokaryotes and lower eukaryotes, requires this bivalent metal cofactor. There are three types of Class 1 ADALases. Type A, which is a major form, is found in the muscles. Type B is in liver and kidneys, and type C is in the brain. A little bit about the history of this. In 1934, ADALase was found to be present in muscle and yeast. Muscle ADALase was first separated from isomerase in 1940, and it was first proposed the presence of ADALase in bacteria in 1934, which was later confirmed in this E. coli. In the 1950s and 60s, the reaction mechanisms was investigated, and the classification scheme that divided ADALase into Class 1 and Class 2 was developed. Work began investigating the inhibitors began in the mid-1960s and early 1970s, and in 1969, the crystals of ADALase were studied in order to determine the tetrameric subunit structure. The first low-resolution crystal structure was determined in 1985 and was refined in 1987. Continuing on with the history of this, then after some debate over the mechanism and the involvement of particular active site residues, it was determined that in 1993 that a residue was not present at or near the substrate binding site despite initial studies, including otherwise. The first high-resolution structure of rabbit ADALase was reported in 1997. Today, work is continuing on ADALase inhibitors. Studies are also being conducted to better understand its utility, the chemoenzymatic synthesis, its interactions with other cellular proteins, and its reaction intermediates. Specificity. ADALase is highly specific for its substrate D-fructose. Now we're talking about the composition of this and the molecular structures. For the composition, the three isozymes A, B, and C found in mammals are composed of four identical subunits. E is a polypeptide chain of 360 amino acids. The enzyme has a homotetrameric structure with subunits that are approximately 40,000 Daltons in size. The structure of these subunits is a beta barrel surrounded by alpha helices and connecting loops. If activity of one of the four subunits of the homotetramer is knocked out, the entire complex is incapable of normal function, indicating a tight communication between these active sites. And in the molecular structure, the primary structures of Class 1 eukaryotic enzymes are highly covered. However, there are no similarities between the sequences of Class 1 and Class 2 enzymes, as they have separate evolutionary origins. However, both Class 1 and Class 2 enzymes contain carboxy-terminal tyrosine residues that are required for maximal catalytic activity. Prolonged contractile activity reduces the type A ADALase expression in messenger RNA by downregulating cytoplasmic glycolytic enzymes and enhancing mitochondrial enzymatic activity. Now we're going to actually start talking about the actual differences in the three types of these ADALase. Aldo A, or aldolase A, also known as fructose bisphosphate ADALase, is an enzyme that in humans is encoded by the Aldo A gene by chromosome 16. The protein encoded by this gene is a glycolytic enzyme that catalyzes the reversible conversion of fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Three ADALase isozymes, A, B, and C, encoded by three different genes, are differentially expressed during development. Aldolase A is found in a developing embryo, is produced in even greater amounts in adult muscle. Aldolase A expression is repressed in the human liver, kidney, and intestine, and similar to aldolase AC levels in brain and other nervous tissue. Aldolase A deficiency has been associated with myopathy and hemolytic anemia. Alternative splicing and alternative promoter usage results in multiple transcript variants. Related pseudogenes have been identified on chromosomes 3 and 10. Continuing on with aldolase A. Aldolase A is one of the glycolytic enzymes primarily found in a developing embryo and adult muscle. Recently, a new role of aldolase in several cancers have been proposed. However, the underlying mechanism remains obscure and inconsistent. In this study, it was found that by investigating aldolase-associated genes using available microarray datasets to help elucidating the role of aldolase in cancers. Aldolase could contribute to the progress of cancer at least partially through its association with genes relevant to cell cycle independent of glycolysis. These genes plus aldolase represents a potential new signature for development and prognosis in several cancers. Metabolic functions is a hallmark of cardiac hypertrophy and heart function failure. During cardiac failure and metabolism of cardiomyocyte switches from fatty acid oxidation to glycolysis. However, the roles of these key metabolic enzymes in cardiac hypertrophy are not fully understood. Here in the present work, it was found that identified that ALDO-A as a core regulator of cardiac hypertrophy. The messenger RNA and protein levels of ALDO-A were significantly regulated in transverse aortic constriction, and the isoproterenol induced hypertrophic mouse hearts. Overexpression of ALDO-A in cardiomyocytes promoted ISO-induced cardiomyocyte hypertrophy. However, ALDO-A knockdown repressed cardiomyocyte hypertrophy. In addition, adeno-associated virus 9 mediated in vivo knockdown of ALDO-A in the hearts rescued ISO-induced decreased in cardiac ejection fraction and fractional shortening and repressed cardiac hypertrophy. Mechanism study revealed that ALDO-A repressed the activation of AMP-dependent protein kinase signaling in a liver kinase B1 dependent and independent manner. Inactivation is a core mechanism underlying ALDO-A-mediated promotion of ISO-induced cardiomyocyte hypertrophy. In contrast, activation of this with metformin blocked ALDO-A function during this as well. Now we'll go to ALDO-B, aldolase B. Aldolase B is also known as fructose bisphosphate aldolase B, or liver-type aldolase, is one of the three isozymes, A, B, and C, of the class 1 fructose 1,6-bisphosphate aldolase enzyme, and plays a key role in both glycolysis and gluconeogenesis. Generic fructose 1,6-bisphosphate aldolase enzyme catalyzes the reversible cleavage of fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, as well as the reversible cleavage of fructose 1-phosphate into glyceraldehyde and dihydroxyacetone phosphates. In mammals, aldolase B is preferentially expressed in the liver, while aldolase A is expressed in muscle and erythrocytes, and aldolase C is expressed in the brain. Slight differences in isosome structure resulted in different activities for the two substrate molecules, FBP and fructose 1-phosphate. Aldolase B exhibits no preference and thus catalyzes both reactions, while aldolases A and C prefer FBP. In humans, aldolase B is encoded by the ALDO-B gene located on chromosome 9. The gene is 14,500 base pairs long and contains 9 exons. Defects in this gene have been identified as the cause of hereditary fructose intolerance. Continuing on with ALDO-B. ALDO-B, the rise in the fructose consumption and its correlation with symptoms of metabolic syndrome has highlighted the need for a better understanding of fructose metabolism. To that end, valid rodent models reflecting the same metabolism as in humans, both biochemically and physiologically, are critical. A key to understanding any type of metabolism comes from the study of the disease states that affect such metabolism. A serious defect of fructose metabolism is the autosomal recessive condition called hereditary fructose intolerance, caused by the mutations in the human ALDO-B gene. Those afflicted with this experience liver and kidney dysfunction after fructose consumption, which can lead to death, particularly during infancy. With very low levels of fructose exposure, these patients develop non-alkali fatty acid liver disease and fibrosis. Sharing liver pathology is also seen in the MBS. Continuing on with ALDO-B, deregulation of ALDO-B has been reported in hepatocellular carcinoma. However, its clinical significance and its role in pathogenesis remains largely unknown. The downregulation of ALDO-B could indicate a poor prognosis for HCC patients, and therefore ALDO-B might be considered a prognostic biomarker for HCC, especially at the early stage. In addition, ALDO-B inhibits the invasive features of cell lines, partly through the TET1 expression. Now we're going to go on to ALDO-C. ALDO-C is also known as fructose bisphosphate. ALDO-C is an enzyme that in humans is encoded by the ALDO-C gene on chromosome 17. This gene encodes a member of the cell, a class 1 fructose bisphosphate aldolase gene family expressed specifically in the hippocampus and Purkinje cells of the brain. The encoded protein is a glycolytic enzyme that catalyzes the reversible aldo cleavage of fructose 1,6-bisphosphate and fructose 1-phosphate-2 dihydroxyacetone phosphate and either glyceraldehyde 3-phosphate or glyceraldehyde, respectively. The amino acid sequence of ALDO-C is highly similar to those of the other isosomes, sharing a 68% identity with ALDO-B and 78% identity with ALDO-A. And they are conserved in the active sites of the three isosomes. This active site is located in the center of the homotetrameric alpha-beta barrel structure of these aldolases. However, several structural details set ALDO-C apart. For instance, some of these adopt an intermediate conformation between the liganded and unliganded structures observed in the other isosomes. Also, the C-terminal region between these some form a salt bridge with the barrel region that is absent in the A and B isoforms. Moreover, the electrostatic surface of the ALDO-C is more negatively charged, which may serve as an acidic binding site or as a docking site to accommodate a C-terminal conformation. Continuing on with ALDO-C. ALDO-C has long been used as a marker for the study of cerebellar compartmentalization due to its specific expression in distinct subpopulations of cerebellar Purkinje cells that are arranged in functionally significant longitudinal stripe-shaped compartments. Each longitudinal compartment determined by the striped ALDO-C expression patterns in its innervated by climbing fibers originating from a specific subarea of the inferior olive and mossy fibers of different sources to a certain extent. PCSs in each ALDO-C compartment then project to a specific subarea of the cerebellar nuclei. In addition, expression patterns of many molecules are closely related to that of the ALDO-C in these PC populations. For example, phospholipase CB4 is expressed in a complementary pattern of that of ALDO-C. Moreover, PCs in ALDO-C positive and negative compartments have different physiological properties. These features of ALDO-C compartments are generally consistent between individuals and are preserved across mammalian species from rodents to primates. Thus, ALDO-C compartments appear to reflect a basic organization of the cerebellar cortex. Okay, now I have about a little over a minute on this first video, so what I'm going to do is I'm going to flip through and let you see what's going to be going on after we finish this here. On our second part, this was part 1. Part 2 we'll be talking about like I said, the African clawed frog. We'll be covering the expression of all those of how it's expressed throughout that one specific species. We'll cover the conclusion, and then I'll cover the references that I'm using on this as well. So basically, that's the first video. I hope everybody has learned something from at least the first part of that, and please join me for this second part on this presentation. We'll be starting at slide 11. Thank you very much.